A small permanent pond is a habitat for a climax community of producers, aquatic plants and algae, and consumers, bacteria, protista, worms, snails, arthropods, and small vertebrates like newts and fish. Why might ecologists call this a climax community? Well, it's the final community. It's stable now. So no further succession. The productist, Paramecium caudatum, is usually between 200 and 300 micrometers in length. An accurate measurement would help in the correct identification of a specimen from this pond. What laboratory equipment would you select to make an accurate measurement of the length of Paramecium caudatum? Well, you'd be using a light microscope, because of the size of it, and so that you can measure it, you would use your graticule. An animal fell into the pond, it drowned and decayed. Within a year, the biological compounds in its body had been completely recycled. What nitrogenous excretory, so it's nitrogenous, it's excretory molecule from the decomposers would pass to the next stage of the nitrogen cycle. So we know that nitrogenous excretory molecule would be urea. Complete the flowchart to show what happens to this nitrogenous compound and name the groups of bacteria involved at steps 1 and 2 as it is converted to a form that plants can take up and use. So we're going to go from the way that it can be taken up is a nitrate. So the stage in between is nitrite, and what goes from nitrogenous compound to nitrite is nitrosomonas. Bacteria, Somonas, and Nitrobacter. So identify 2 features of this cell that confirm it is not a prokaryotic cell. In each case, state the letter and the name of the feature. So we're looking for organelles which are bound by a membrane. So there's A for rough ER. There's D for the Golgi apparatus or the Golgi body. Also there's E, which is the secretory vesicle. And you could also have F, which is the mitochondrion. The cell is capable of synthesizing and secreting proteins. Using only the letters, list the correct sequence of the organelles involved in synthesis and secretion of a protein. So we want C or A, the rough endoplasmic reticulum. Then D, which is the Golgi apparatus, where the proteins are packaged up and processed. And then E, where they are secreted through the secretory vesicle. So that's 3 marks. Peroxisomes are vesicles that usually contain enzymes such as catalase. Explain how peroxisomes can be moved around the cell. So now we're talking about the cytoskeleton, aren't we? So they need to attach to the cytoskeleton. Cytocell skeleton structure. And these change in length. So move it around. So, and they change in length by using protein motors. So catalase is an intracellular enzyme with an iron-containing heme group. State the term used to describe an ion that is essential for the enzyme to function. So this is an extra part of the enzyme, so it could be called a prosthetic group. And it's inorganic, so it's a cofactor. Name another conjugated protein that contains a heme group. Well, the obvious one that I always teach is the hemoglobin, because it's a great example of an enzyme with its quaternary structure, its conjugated protein, its heme group. It's great. Figure 20.2 shows the effect of copper ions on the activity of catalase. What can you conclude from this graph about the type of inhibition shown? Explain your answer. Well, if you look at this straight away, it just drops, doesn't it, as soon as the copper ions are added. And no matter how much more the concentration of the hydrogen peroxide, which is the substrate, is added, it still doesn't increase, it just stays the same. So this suggests that it's non-competitive. So it's attaching to another part of the enzyme and therefore changing the shape of it, so therefore the hydrogen peroxide can no longer attach to the active site. So it's non-competitive. The rate of reaction does not increase as the concentration of substrate increases. Right, so now we're on to a question about beavers. Now, these are relatively current in the news as well, because there have been some released on the River Otter relatively recently, and there's some being released in Derbyshire as well. These ones are ones that were recently introduced into Scotland and hailed as a great success after researchers found that the animals had transformed the landscape after 5 years that beavers had constructed dams, the largest of which was 18 metres long and 1.6 metres high, felled trees, created canals, built lodges, which are their large nests, and successfully reproduced. Beavers are considered to be a keystone species. Explain why they are a keystone species in their native Canada. So they have a significant effect on the ecosystem. And so therefore, if they were removed, it would unbalance the ecosystem, because many of the other species rely on them for survival. So it is really important that they stay in that ecosystem. Right, next, one benefit of the reintroduction of beavers in Scotland was an increase in habitat diversity. Explain how the following activities could have contributed to increased habitat diversity. Right, so constructed dams, that's obviously going to lead to flooding. So it floods upstream, but downstream it's going to slow the flow rate of the stream. So upstream you get flooding, and downstream you get a slowed rate of flow of water. Now, when rivers and streams run very quickly, it's hard for many species to live in them, because they just get washed downwards and they can't survive, they can't find somewhere to live, they can't find food. So this slowing of the movement of the water means that it makes it an easier environment for these aquatic species to live in. So felled trees then, well, as soon as you fell some of the trees in a wood, you're opening up the canopy, and it's been shown that this increases biodiversity. So a lot of woods, we now have some cutting down, felling of carefully chosen trees to open up the canopy. So the felled trees mean that we get an opening up of the canopy, so more sunlight can get to the species on the floor of the forest or wood, allowing more light so they can photosynthesize more, or and so that if you can have more photosynthesis of plants, and that means that there's going to be more species living on them, more herbivores, etc. So the building of lodges then, this is going to create habitats for, and sheltered habitats for insect species. So provides sheltered habitat for insects, just like building an insect hotel in your garden. Basically, that is what beavers are producing. Suggest one other benefit of the reintroduction of beavers, where there's quite a few examples of this. For example, where they're introducing them near me, then there's scientific study and schools are allowed to go in. So scientific study. They also try to encourage tourists in to look at them, so it's ecotourism. Education. It could improve the water quality, because when the dams are built, then it stops the water from flowing quite so quickly, and so there's less silt carried in the water. So better quality. Okay, so increasing habitat diversity may lead to an increase in species diversity and genetic diversity. Explain why species diversity and genetic diversity may be increased as a result of the beaver's activity. So if there's more species able to live in the area due to the effects of the beavers, such as a slowing of the water, the building of their dens, their lodges for the insects, the opening of light down to the forest floor, then the more species able to live in an area, then that gives you greater species diversity. And as the species have a wider range of conditions in which to live, then that increases the genetic diversity. As it allows for adaptation, etc. And therefore adaptation and survival. Just what all species need. But not all people are happy about the reintroduction of beavers. Suggest two arguments that may be used by local business leaders against the introduction of beavers, and state whether these outweigh the arguments presented by the naturalists. Right, well, flooding is an obvious one, because you get the loss of the farmland. And I can understand why a farmer might be upset about that, because it's his land or her land. But the strength of the argument depends on the quality of the land affected. If it was just laid to fallow, for example, then they haven't really got much to say about it. But if it was like really good pasture land or really good growing where they can grow an expensive crop, then yeah, they've probably got a stronger argument. All right. But to be fair, it tends to only affect small areas. So again, maybe not a great argument. Trees being cut down. The foresters are unhappy about trees being cut down. But this is only likely to occur by the water. as the beavers are not going to want to carry or drag the wood long distances. So it only tends to happen down by the water. So it's not a strong argument. But there could be damage to the riverbank, and if there's eroding maybe to riverbank, and that could be expensive to repair, especially if there's erosion and buildings near the bank. But basically they could say that costs should be covered by those benefiting. By the beaver present. So now we'll move on to some genetics. The sweet pea has been used to study inheritance since the 19th century by good old Gregor Mendel. The seeds of the sweet pea can vary in colour and shape. The gene that controls colour has two alleles. The dominant one is yellow and the recessive is green. A gene that controls shape has two alleles. The capital R, dominant one, produces round seeds, but the recessive one produces wrinkled. In the 19th century, Gregor Mendel crossed a pea plant that was heterozygous for both seed colour and shape with a pea plant that had green and wrinkled seeds. List the gametes that would be produced by a sweet pea plant that is heterozygous for both seed colour and shape. So if it's heterozygous, we're going to be looking at YYRR. So there's 4 different gametes that can be produced here. We can have a capital Y and a capital R. And we put a little circle around to show that it's a gamete. Capital Y, little r. Little y, big R. And little y, little r. There we go. So list the genotypes of the offspring that were produced from Mendel's cross and state the corresponding phenotypes. So he crossed a heterozygous with a homozygous recessive. So that's the cross. So we have big Y, big R, big Y, little r, little y, big R, little y, little r. And all you can have from the homozygous recessive is that. So what have we got? We have big Y, big R, little r. Big Y, little r, little r. Little y, big R, little r. Little y, little r, little r. So what do we have? Well, we have yellow and round. We have yellow and wrinkled. And we have green and round. And we have green and wrinkled. Now for a bit of mathematics. When Mendel crossed 2 pea plants that were heterozygous for both seed colour and shape, the ratio of the phenotypes in the offspring was your classic 9:3 to 3:1 ratio. Some students tried to recreate this investigation using a modern variety of plant that showed the same phenotypic ratio in seed colour and shape. The students crossed 2 of the modern plants that were heterozygous for both seed colour and shape. The results of the cross were this: 58 yellow and round, 31 green and round, 21 yellow and wrinkled, 2 green and wrinkled. So first of all, if we're going to have a look at this, we're going to be looking at using the chi-squared formula to compare their data to the expected 9:3 to 3:1 ratio. So what we have to use is we're going to be using the observed, so that's the observed, in case you've forgotten, and E is the expected. So this is obviously the observed. I've already put it into the table for you. So to find the expected, what you would have to do is add these up. So we've got 23, 53, 54, 112. And those are in the 9:3 to 3:1 ratio. So 9, 10, 11, 12, 13, 14, 15, 16. So 16 into 112 gives us 7. So this would have 7. This would have 3 times 7, which is 21. 3 times 7 is 21. And 9 times 7 is 63. So those numbers go into the expected. So now we've got to look at this area up here, the O minus E squared. Well, we'll start with O minus E, makes it easier. So O minus E first. So all you do is you take the biggest number, the smallest number from the biggest number. So 63 minus 58 is 5. 31 minus 21 is 10. 21 minus 21 is 0. And 7 minus 2 is 5. So now we can square it and put it over the expected. So this is 5 times 5 is 25. Over the expected here, which is 63. Here it's 10 times 10, which gives you 100. And then over the expected is 21. Here, 0 squared is 0, over 21 is 0. And this one is 5 times 5 is 25. And then we put it over the expected, 7. So you then just calculate each of those out. And we then add them all up because that's what that sign means. That means to find the sum of. So if you add all of those numbers up, the 25 divided by 63, the 100 divided by 21, and the 25 divided by 7, you end up with 8.73. So remembering it was 8.73, what we need to do now is, after analysing the results, the students stated that the inheritance of the seed colour and the shape in their investigation was different from that in Mendel's investigation. Using table 17, discuss whether the results of the investigation and the chi-squared test support the student's statement that it was different from Mendel's investigation. So we need to look at the probability of 5% chance that the difference is due to chance, or the 95% chance that it isn't due to chance. So we tend to use the 0.05, which is 5%. And then the critical value, because we got 4 results, and so the number of results minus 1 is 7.82. So the critical value from the table equals 7.82. And our number is higher, is greater than 7.82. So this means that there's less than 5% chance that difference is due to chance. Therefore, the results are significant. A ratio that is different from the expected 9:3 to 3:1 ratio in a cross such as this can be the result of epistasis. Suggest and explain one reason other than epistasis why the phenotype ratio might not be 9:3 to 3:1 ratio. So basically you could say this is to do with autosomal or just linkage, where both alleles are on the same chromosome. So therefore, there's no independent assortment. And so the alleles are inherited together. As they are both in the same gamete. Of course, sometimes that doesn't happen. If you've got linkage, that can still be changed because of chiasmata or crossing over. The yellow colour in peas is the result of an enzyme that breaks down chlorophyll, which is green. The dominant allele codes for the production of an enzyme that breaks down chlorophyll. So that's the dominant one. The recessive allele is a result of a mutation in the dominant allele. The recessive allele codes for an inactive form of this enzyme, so there won't be breaking down of the chlorophyll. Outline how the dominant allele codes for the production of this enzyme, and explain why the recessive allele codes for an enzyme with a different primary structure. So this is all about the genetic code being transcribed and translated into an enzyme at the ribosomes, how that occurs, and how you can end up with mutations which can change the enzyme produced. So we need to talk about the DNA code, which has got your 4 nitrogenous bases working in triplets or working in codons, and 1 codon codes for 1 amino acid. And it's a degenerative code, so not all mutations are going to affect which amino acid goes where in the protein. You need to talk about the fact that messenger RNA copies the code, transcription, in the nucleus. That transcription occurs due to complementary base pairing. Allowing the synthesis of messenger RNA, helped by RNA polymerase. The messenger RNA then moves out of the nuclear pore, through the nuclear pore, into the cytoplasm, and attaches onto ribosomal RNA, the ribosomes, in the cytoplasm. And now translation is going to occur. So you've got the messenger RNA is bound to the ribosome, or the ribosomal RNA, and then transfer RNA brings the correct amino acid into the correct codon, as the ribosome reads the code. And so this builds into your polypeptide, which forms your enzyme. However, mutation, either when the DNA is being copied or when the messenger RNA is being formed, mutation can lead to a difference in the primary structure of the protein due to the wrong amino acid being coded for. And this can happen due to substitution, deletion, frame shift, okay. This is an important question. This is one of those ones where you get the extra marks for writing it out into a nice, clear explanation. With reference to the proteins coded for by the seed colour gene, explain why the recessive allele is recessive. This is because the active enzyme In other words, the dominant one from the dominant allele will still be synthesized even if there is only one dominant allele. It will still be expressed. Next then, a student investigated the distribution of the Buttercups ranunculus bulbosus in a field which contained a pond. The student noticed that Buttercups near the pond looked slightly different from those further away. After further investigation, the student identified the Buttercups near the pond as a different species, Ranunculus repens. Describe how the student would use a belt transect to investigate the distribution of the two Buttercup species. So here's your pond. There we go. And what you would do is you would lay down a tape measure. And then at regular intervals along that tape measure, you would place your quadrats. Then obviously you have to identify the type, the species of Buttercup in each quadrat and count the number of each species plant in each quadrat and record it. You could say percentage cover, but they're relatively easy to count. So there we go. Use the space below to show the format of a results table suitable for recording the results of the student's investigation. You do not need to include any suggested data. So you are choosing to do quadrats at certain distances up from the pond. So your independent variable equals distance from the pond. So that goes in your first column. The dependent variable equals the number of each species. So those go in the other column. The other thing you need to remember are units go in the column heading. So we're obviously going to have one. Okay. I would go for distance from pond. And so that would have units of meters. And then ranunculus bulbus, bulbulus, or whatever it is, lus, and ranunculus repens go in there. And that's the number of, so we know what we're measuring there. And you'd obviously have at least 10 quadrats. 3, 4, 5, 1, 2. Obviously you would use a ruler. 3, 4, 5. As the ground dipped towards the pond, the soil became obviously wetter. The student thought that the soil moisture might affect the distribution of the two Buttercup species. Suggest one biotic. So this is abiotic, isn't it? Because it's not living. Suggest one biotic factor that might affect the distribution of the Buttercups. Well, I think the most obvious thing is it being eaten. Herbivory, or just being eaten is fine. Or there could be competition for space, light, etc. As an extension to the fieldwork, the student decided to investigate the effect of soil moisture on the germination of Buttercup seeds. So germination of Buttercup seeds. The student planted ranunculus bulbosus seeds into 5 pots and maintained the soil moisture in each pot at different levels. So this is going to be your independent variable, isn't it? 0%, 20%, 40, 60 and 80. This was repeated with seeds from ranunculus repens. After 4 weeks, the student counted the number of plants that had germinated in each pot. Table 16 shows the results of the student's investigation. State 2 factors that could be controlled as part of this investigation. So controlling. They haven't said how many seeds, so the number of seeds in each pot should be the same. The type of soil or compost should be the same. The mass of soil or compost. Temperature is one that everybody comes out with. Those are the obvious ones. It could be oxygen concentration. Light intensity doesn't normally have any sway on germination because they are under the soil and they're not getting light there anyway. And so we come to the last question, and a paper would not be complete without a graph. So use the grid provided to plot the results of the investigation. So first of all, I want you to think of St. Paul scale, title, plots, axes, units, lines. As long as you get all of that, we should get full marks. So first of all, you need to think about the scale. So soil moisture is obviously going to go across the bottom because, as I said, the independent variable is the soil moisture, and that's the basis of the experiment. So if it's the base of the experiment, it goes across the bottom here. We're going from 0 to 80. Now, obviously, you would be using a ruler and a sharp pencil. So 20, 40, 60, 80. They normally give you paper the right size. I had to cut this one down to fit it on to my slide, though, so I'm going to have problems going up the side. So 60, 80. So this is just the scale at the moment. I'm not worrying about anything else. So here we go up the side. And what's the biggest number? 46. That's going to be a pest, isn't it? 10, 20, 30, 40, 50. That's as good as I'm going to be able to go, I think. Yeah. Okay, so we're going to have a sensible scale. So 10, 20, 30, 40, 50. And it starts at 0. So now we're going to, so we've thought about the scale. Now we've got to think, and we've got the axes the correct way round, but we need to label them. So this is soil moisture. And this is the number of seedlings germinated. So we've already got the units there because it's number. So we've done the axes, but we need the units for here, which is %. So we've got that. So now we want to think about the plots. So it's continual data, number against number, so I would choose a line graph. So for the first one, I'll do it in red. So ranunculus bulbosus, 0, then 23, which is there, if I can see it. Better put my glasses on. 0, and then 0, 23, then 46, so that's 46 up there, and then it's 16, so that's to there, and then it's 4, so that goes to there, and so we have a smooth line like that, as smooth as I could do using my pencil on here without touching everything else. So that's that one. The next one, the ranunculus repens, starts again at 0, then it goes to 8, which is that one, then it goes to 15, and then it goes to 42. It definitely likes the wetter soil, and then it goes to 33, where it might have just got a little bit too wet. So here we go. So the green is ranunculus repens, so we need to have our key, don't forget, so we know which line is which. And this one is ranunculus bulbosus. So we've done the plots, we've done our lines and graph to show how soil moisture affects germination of seeds in Ranunculus bulbosus and Ranunculus repens. And so we finish.